So let's talk about question number 67. Question number 67. So when you talk about question number 67, it is basically a very simple question, and it is from environmental biotechnology, which is a part of your third module, and have been very well covered by Tanya ma'am, right? And there we talked about plasmid, phase mid, right? What are restriction endonuclease and how the plasmid work, right? So let's talk about question number 67. So when you talk about question number 67, it's very simple question, and we go by statement. So given below are two statements, statement 1 and 2, and then we have to see in light of the above statement, the choose the most appropriate answer from the option given below. So let's see both the statement what they says. So statement 1 says what? Statement 1 says that many bacteria carry plasmid. So plasmid is what? It is basically what it is? Covalently closed circular DNA. It is basically covalently closed circular DNA, yeah, extra chromosomal in nature, right? It is covalently closed circular DNA, right? Nature कैसा होता है? Extra chromosomal, right? Extra chromosomal. This is not a part of the chromosomal DNA, that you have to keep in mind, right? So this much information you should be having in mind, because it is mentioned well in that module also, right? Then the second thing which they are mentioning here, which are much smaller pieces of DNA, also circular, right? As I told you, and self replicating. Obvious सी बात है कि ये भी DNA replication show करता है, क्योंकि plasmid जो है replicate तो करेगा, that is the basic property. Okay, then as per the things given here, this statement is true. So statement 1 is true. Now let's come to the question, statement second. Statement second says that plasmid are vitally important in the context of environmental biotechnology, in that they frequently carry the gene for degradative pathway. This is very important. Whatever biodegradation happens, or whatever bioremediation happen through microbes, this thing have to be kept in mind, that those microbes contain the plasmid for the degradation. For example, if you talk about Pseudomonas putida, which the question come in 2019, so that superbug designed by Anand Mohan Chakravarti was having plasmid for the degradation of benzene, toluene, ethylene and xylene. So 4 substances it can degrade. So these plasmid are vitally important. Yes, they are very important when it comes to environmental biotechnology, because they carry the gene for the degradation pathway. So these plasmid have those gene for a particular degradation pathway. That's why this statement is also correct. So both the statement here are true. So the correct option here is 1, because both the statement given in the question are true. So first would be the correct answer for 67. Is it clear to everyone? Anyone having any doubt in this? Jaldi se boliye bhai, fatafat se, quick and fast. Understanding the things is very important, because that will keep your level up in comparison to other. Clear? Anyone having any doubt till here? चलिए 67 के बाद next question पे चलते हैं. If we have certain, so we have mentioned here, see, you can see here what plasmid is, how they are carried out, everything have been mentioned here. So plasmid is a small circular piece of DNA. Yeah, so you can see here, and this is the plasmid pieces which are there. Then functions of plasmid, you can see they are quite important. Then type of plasmid, then specific type of plasmid. Yeah, these are the plasmid, like degradative plasmid, which this question is talking about. So it is a one sort of plasmid which is called as degradative plasmids. So degradative plasmid basically help the host bacterium to digest the compound. They are not commonly found in nature, such as camphor, xylene, toluene, salicylic acid. And these plasmids contain gene for specialized enzyme and break down specific compound. Degradative plasmid are conjugative. So this is the question which is there. So I hope this question number 67 is clear to everyone. Right, now we are moving toward question number 68.